catch up a little because we're down 10 minutes.
Oh, okay. All right. Hi, we are NanoMacro, and we are working on a platform to develop in situ immuno-oncology.
What is in situ? Please define your acronyms for me, please.
We will explain shortly. First, I'm Alec Plotkin.
Yeah.
I'm Michael Gianni.
I'm you.
And I'm Abdul Hussein.
Got you down. Yep. Thanks.
So, in recent years, we have seen an explosion of immuno-oncology therapies. Immuno-oncology utilizes the body's own immune system to fight cancer. And what we are seeking to do is to apply treatment strategies for these therapies in situ, so actually in the body. One of the most successful immuno-oncology approaches so far is engineered cell therapy. Engineered cell therapy takes white blood cells from the patient's own tumor microenvironment, removes them, and edits them to express a cancer-fighting modality. The most successful of these is called the CAR T cell, which stands for the chimeric antigen receptor. This approach has been extraordinarily successful to date, showing remission rates of up to 83% in some forms of leukemia. However, the technology still has a ways to go. It has trouble penetrating solid tumors, unlike leukemia, which is a blood cancer. Also, it exhibits high production costs because the process of producing CAR T cells is extremely labor intensive, involving isolating white blood cells from the patient, then taking them into a lab, engineering them to express a chimeric antigen receptor, and finally putting them back into the patient. So it's individualized care, but more research and new approaches will be needed in order to make this technology commercially viable on a wide scale. Our NanoMacro's big idea is to deliver gene-edited components to engineer these immune cells in vivo or in situ on the site of cancer. We will use nanoparticles as the drug delivery vector for this approach due to their ability to end up in tumors by the so-called EPR effect. The EPR effect, or the enhanced permeability and retention effect, is a result of the fact that nanoparticles cause drugs to circulate for longer before being taken up by cancer cells. The cancer architecture tends to have leaky cell vasculature, leaky vessel vasculature, which is a result of aberrant cell reproduction in the tumor. So nanoparticles are able to seep out into the surrounding tissues and enter the tumor cells that are their target. The nanoparticles that we will be using have a chitosan shell, which is an ideal environment for encasing nucleic acids, which are generally negatively charged, a nucleic acid interior, which contains our gene editing components, and a hyaluronic acid coating, which allows us to target specific cells within the tumor. So, among the nucleic acids in the interior of the nanoparticles, we'll have an mRNA or messenger RNA for a CRISPR-Cas9 gene editing machinery. We'll have a guide RNA to target a specific gene that we want to take out of our target cells, and we'll have potentially even DNA for an extra gene that we're going to add in that will target the specific tumor cells that we want to create an immune response against. So, instead of going directly for the cancer cells, we'll be targeting macrophages, which are a component of the immune system in a separate class from white blood cells like T cells and B cells, which are lymphocytes. The reason that we're going for macrophages is because they're ubiquitous in the tumor microenvironment. They easily infiltrate this environment due to the fact that they're recruited by the tumor itself to allow it to prolong its life cycle. So the tumor will secrete cytokines, which are like signaling molecules that tell the macrophages to turn down the immune response, to secrete other molecules that tell T cells, B cells, etc. not to go into the tumor. And this is, in essence, an anti-inflammatory response. In other words, this is known as M2 polarization, and we want to reverse this effect. Tumor-associated macrophages, in addition to being the arbiter, the kind of gatekeeper for this response, are also an easy target for nanoparticles. CD44, which is a cell receptor that hyaluronic acid binds to, is highly expressed, most highly expressed even, in monocyte and macrophage lineages. Also, this type of cell tends to internalize nanoparticles in the 10 to 100 nanometer range, which has been shown again and again in prior research. So a quick overview of the gene editing therapy that we'll be putting in the cells. This uses CRISPR, which is a groundbreaking new method for altering the genomes of cells. So our goal is to eliminate the pro-tumor and anti-inflammatory response and to turn it around so that we can attract immune cells to the tumor and negate some of the prolonging life cycle effects that the tumor has. We want to basically get the body to attack the tumor and put it into regression. So one of our targets in this approach is a gene that in the macrophage that causes the immune cells to stay away, that causes an anti-inflammatory response. So what we want to do is, in our nanoparticle, we'll have the mRNA for the Cas9 protein, which is what actually goes around to the DNA and figures out where to take this gene out. The Cas9 will complex with our guide RNA, which tells it where in the genome to target, and then after engaging the DNA, it will recognize a very specific sequence and create a cut. We need 2 Cas9s and 2 gRNAs in order to knock out the entire gene, and these 2 ends will be joined together through a process called non-homologous end joining repair, and this gene will no longer exist. The idea of this is to get rid of one particular target. In this case, there are several possible culprits we could go after, but the one that may show most promise is called MERTK. This gene is a member of the TAM family, which also includes genes like TYRO3 and AXL, and it binds tumor-secreted anti-inflammatory cytokines, one of which is Gas6. These, as I have been saying, downregulate the immune response and cause the macrophage to release anti-inflammatory cytokines which keep away other cells. So, by eliminating the MERTK gene, we hope to reverse the so-called M2 polarization, which creates this anti-inflammatory response, and through recycling of the cell membrane, eventually the receptor that we're trying to target will disappear, and the TAMs, essentially the tumor-associated macrophages, essentially become double agents, in that they actively infiltrate the tumor cells and are recruited by the tumor, but we can now program them to create a positive immune response and conjugate it with other chemotherapies, other immunotherapies, in order to send the tumor into remission, hopefully.
The M2, is that there are 2 major types of macrophages, and M2 is the anti-inflammatory, M1 is the killer.
Yep.
Is that what you mean?
Exactly. So we want to basically convert M2 macrophages into an M1-like phenotype. And now we'll take you through the business case.
Yeah, so the business case for immuno-oncology is very large. The estimated market size by 2021 is $95.8 billion to $119.39 billion. CAR T therapy is just one component of immuno-oncology. There are many competitors, including Shire Pharmaceuticals, Pfizer, Bristol Myers Squibb, Novartis, and there's currently only one company in CAR macrophages. For drug delivery systems, they represent a multi-billion-dollar industry. So drug delivery systems are something that has been looked at for the last decade, but hasn't really been keyed in on or focused on. One important aspect of drug delivery systems is their ability to limit, when done right, their ability to limit the symptoms that a patient gets from using certain medicines or drugs. The most common drug delivery systems that you've probably heard of are local chemotherapy, polymer drug conjugates, liposomal systems, and transdermal delivery patches. Each idea between each drug delivery system is to improve the efficacy of each drug whilst minimizing the symptoms or side effects. The most common example of a drug delivery system would be an oral drug. For example, seasonal allergy drug. And most of the side effects that come with seasonal allergy drugs include drowsiness and upset stomach. And basically the advent of drug delivery systems is to try to minimize those side effects while keeping the efficacy of the drug. So drug delivery systems, especially when partnered with polymers, the diffusion of such large molecules through polymers are conventional controlled release systems. So basically, they increase the ability of the drug to diffuse into the body. Polymers oftentimes can be targeted specifically to locations of the body, so they have higher targeting rates, and they also have something called stealth mechanisms, so the ability to trick the body into not knowing that they're there. One example of this is polyethylene glycol, which I'll talk about later. And many venture capitalist companies are looking for biopharmaceuticals that are working with nanopolymers. For example, Flagpoint Pioneers, venture capitals. Flagship Pioneers, yeah. So according to the National Cancer Institute, the estimate of 22,850 patients were diagnosed with brain cancer in the U.S. in 2015. So the drug delivery system for cancer currently is various forms of chemotherapy, various forms of drugs whose side effects leave these patients in very precarious qualities of life. It's very tough to locate the location of the cancer specifically. So few companies working with polymers, I mentioned polyethylene glycol before, which is known for its stealth behavior. So one company that's particularly working with polyethylene glycol, and it's located in Cambridge, Mass, is Bin Biosciences. A few other companies that work with polymers are Calando Pharmaceuticals in California. They work with a polymer called cyclodrexin polymer, and it silences gene expression. And as Alec had mentioned earlier, HA, or hyaluronic acid, is the primary CD44 binding molecule. So CD44 is a gene that's overly expressed in solid tumors. And one of the problems with tumors or cancers is being able to specifically target that location. So with HA, we can specifically target the CD44 in those solid tumors, allowing the medicine to have a greater chance of reaching that specific location. Current modern drugs are basically, they target the whole body, so that's why their side effects are often wide and large. But if you're able to target the specific location of the specific disease, you can incredibly mitigate those side effects.
And you said it, CD44 is overexpressed in macrophages and in solid tumors.
So it's expressed in some solid tumors as well. It's not as universally expressed in tumors as it is in macrophages. But it does allow us to potentially deliver to other targets.
Remind me, before I forget at the end, I have a thought about HA.
Okay.
Actually, I can go back a second. Yeah, so I mentioned a few venture capitalist companies interested in biopharmaceuticals. A few of those are Flagship Pioneering, Third Rock, First Rock Ventures, F Prime, and Orbit.
I just had Flagship come and give a talk to one of my conferences that I co-chair, and Third Rock was founded by the guy that founded the company that brought me out here. Mark Levin was the founder of Third Rock. Sorry, continue. Sorry.
That's cool.
So, as I mentioned, we have Bin Biosciences in Cambridge, Mass. that currently works with polymers. So, what better location for a pilot plant? So, we're gonna work in what's called a cluster location. So, in Cambridge, Mass, there are a slew of pharmaceutical companies in very close proximity. For example, Takeda Pharmaceuticals, Zimmer Biomet, which works in pharmaceuticals a bit, Bin Biosciences, which I've also mentioned. So, those companies are kind of what's called a cluster location. So it's when companies that sort of make the same materials are in close proximity, they can sort of share resources and help each other out. So that's basically the mindset for our location. For our pilot plant, we only require a 400 square floor space. So in the floor space, we're going to have 4 sections. There's going to be administration and processing. The actual plant floor is going to be benchtop testing, which will also be the area for quality testing. And then the last area is going to be storage. So storage space, we plan on being able to share storage space with Bin Biosciences, which already work with polymers, so we plan on being able to space out polymers with them. For the plant space, we'll talk about that, more of that later, and the equipment that will be in that space. Benchtop testing and quality testing is just being able to ensure that the quality of the raw materials and also of the product are up to standard. And administration and processing is just paperwork. So
you have 58 square meters in addition. So this room is about twice the size that you think you require.
This room? No.
400 square feet.
That's a pretty large office space. I don't know. That's what so.
Oh, but that's not enough by any means.
And as you see, what I show here is our detailed process flow diagram. And our 3 main materials are the chitosan, mRNA, and mRNA can be the CRISPR, can be CD44, and can be any other things. And also the HA. And for the ideal model, the HA is outside the whole nanoparticle, and it's like the shell, and then inside the chitosan, and then inside is an mRNA to protect.
HA, chitosan, mRNA.
Yep. And
that's the package.
Yep. And chitosan is a renewable biomaterial and non-toxic, so that's why we choose this material as one of the materials. And the specific celebrate the characteristic of chitosan set the limits in the practical usage, because the chitosan cannot be dissolved in the water, even though it is distilled water, but it can be dissolved in the hydrochloric acid. So that's why we use the hydrochloric acid to dissolve the pharma-grade low molecular weight of chitosan powder. And then the whole solution is precipitation by the sodium hydroxide, and then the sediments, and then it's transferred into the filter to filter out the sediments. And the purified chitosan solution is washed. The purified dry chitosan is washed by the distilled water, and then transferred into another dissolver, because from the second dissolver over there. What?
No cells in any of this here.
Yep. So this one is like the purification, and the next step is to form the nanoparticles.
Okay. Just to give you an idea of.
Yep.
Genzyme's plant is in Austin.
Mm-hmm.
And do you know what? Have you seen it? It's like it looks like a church, and it's like 8 floors, and they put an extension on it. And you know what the total output of that is in 1 year? 1 kilogram.
Yeah.
Okay.
Yeah,
But
because
that was based on cell-based products.
Right. And we're
This is
using
all
nanoparticles.
synthetic.
Yeah.
This is all synthetic.
Yeah. So the aim of the first part
Yeah.
is we want to purify the whole chitosan.
Are you going to get chitosan in its purest state as you can get it? Because it often requires purification after you buy it.
Yeah, we've also looked at using various filters and other means of purifying the chitosan as well. We actually also have them included in this flow diagram.
Cool.
And so the next step, as what I mentioned before, so is to form the cross-linking of chitosan nanoparticle. The purified and dry chitosan is dissolved in the hydrochloric acid again to produce chitosan solution. And the concentration you can see over there of the hydrochloric acid is 1 ml, because we don't want to make the whole solution too acidic. And the solution then is transferred into a reactor to react with tartaric acid water solution. And And the whole environment of the reactor, it should be 4 Celsius. And I didn't show it there, but the environment should be under 6.5 pH. And the pH environment is adjusted by the sodium hydroxide. And the reaction is started at 4 Celsius for 30 minutes. After this final step, this reaction step, the final solution is transferred into another filter. And this filter has a specific pore size at 100 nanometers, because we want our fundamental particles under the 100 nanometers because the because of clearance of the whole body. And the rest of the tritose nanoparticle is purified 7 days, and against molecular water, and also freeze dried. And another.
So I'm thinking you should rush through this a little more because we are running out of time.
Okay. I will just give the rough
It's a
detail.
late start and everything, and other people are.
Okay.
Running,
And to form the tritose and the HA nanoparticle, RNA solution is necessary, because what I mentioned before, RNA solution is the most important part. And the RNA solution is in trace buffer, and the pH environment is 7.5. 7.5, and the buffer salts are removed by the centrifugal filter in order to make the pure nanoparticles in the last. And the dry tritose nanoparticles over there is also dissolved in the hydrochloric acid and in the nucleus-free water. For the HA, it's dissolved in the nucleus-free water, and the concentration of 1.5 mg/ml. To control the final nanoparticle size, the solution, the HA the mRNA solution and the tritose solution goes into the mixer first, and the flow rate of this two is depends on the molar ratio between the amine groups in the tritose and the phosphate groups in the RNA. And for the given volume, the RNA solution, tritose solution is added slowly under stirring. Then the mixture is added in the HA solution with the same volume to form the outside shell. So, and
I should really give you this because I'm with you, so you could point to where things are. But get through this quick because there's a lot more to go, right?
Mm-hmm.
And
Please.
our production is over there.
Yeah. Okay.
And the final production we want to do is 1 kilograms of nanoparticles per year. It's
1
not
or
gonna be
10?
10.
Okay.
Sorry.
Okay, good. Thank you.
Okay, so here are the equipment costs associated with the process flow diagram that you introduced for us. So we calculated these using an equation that we found in our textbook by Towler, which we use for our Capstone class. So the S variable is representative of the size of the equipment. And then the other parameters are just defined by the type of equipment.
What is that 10? Is that 1,000 liters or 10 liters or what?
Yeah, so in this table, the units aren't defined, but they were by liters for the mixing tank and meters squared for like the dryers and the centrifuge. So, yeah. So, our total cost of equipment is about $1 million based on these estimates.
Yeah.
Okay, so our capital investment takes into account a number of different considerations for our facility buildup. So firstly, our ISBL is our inner battery limits, which is associated with the facility buildup itself, the cost of equipment and installation that is associated with that. So the recommended factor for installation using with a process that uses solids and liquids is 3.63, which we use to calculate our ISBL. Our OSBL is our offsite battery limits, which is like infrastructure costs, such as like HVAC system. Since we're a pharmaceutical company with a process and we're going to need a sanitary environment, so having the ability to filter out and keep a sterile environment is very important, and keeping that temperature constant throughout the whole process. So we actually calculated these parameters based off our ISBL. So our offsite battery limit is about 40% of our ISBL. Our engineering construction cost is about 20% of our ISBL, and then we have a contingency plan just in case we need to make some changes to these plans that are unexpected. So,
What's a battery?
It's just, I guess, in term to.
It's the details of the individual costs, probably.
Right.
That's fine.
Yeah, it's just a term that they use to describe, whether it was within the facility costs or something to do with infrastructure. So, yeah.
Okay.
Next slide.
Thank you, Brad.
So here's our cost production associated with our pilot plant. So we're using streams of 200 kilograms per year of our tridacin hyaluronic acid.
Are you saying that hyaluronic acid only costs $130 per kilogram or $130,000?
$130 per kilogram, which we got based on low purity grade hyaluronic acid.
You're going to have to purify.
Yeah, we expect for our pharmaceutical purpose that we would need to buy something a little bit more expensive, more pure.
It gets cheaper HA out of Japan, I think.
Yeah,
$2 or $3 a gram, or something like that.
Yeah, and that's probably more associated with like cosmetic purposes than USP. So yeah, most of our cost comes from our RNA cost, our starting material for our nanoparticles, and also we have nuclease-free water, which we evaluate as a consumable for our process. So these are raw material costs.
I think you're way off on the HA, though.
On the HA, okay. All right.
Okay, excellent. Thank you.
So our fixed cost production is incurred regardless of the production rate. So these are our costs that are just associated with running the plant, day in and day out. We start off by assuming that we're going to have 5 employees at the site. So 2 that can monitor the production, actually at the process floor, and then 2 that are providing insight somewhere out in the office.
I'm commenting along the way. I hope you remember these things. If you take a drug company like Amgen, an employee costs 12 times what you have for one. It costs over twice what you have for one employee. But you're a startup, but you can't get away with an employee for $60,000.
Yeah, so that's just based on the salary, and then we also have a cost associated with like health plan and also other benefits.
Overhead. Okay.
That's taking account for the direct salary overhead.
I got it.
And then we have supervision costs, which would just be like
That's
manager.
You broke it up. Thank you.
Yeah, which we assumed was like 25% of our labor costs.
Yes, you might add 30 to 40% over the salary. It's a good.
Yeah, I think the direct salary overhead is a combination of these two.
Yeah. That's fine.
Yeah, and then we have other costs associated with our production, such as maintenance, property tax, rent, which are all based off of our labor costs. And then actually our biggest cost is research and development, which is going to be 20% of our revenue, which we'll go over on the next few slides. But it's a humongous bulk of our cost, which isn't a bad thing. Yeah, and then we have environmental royalties and interest payments, which are based on our just our facility costs. We've made those estimates.
Well, aren't facilities a direct cost? Where do you have that?
We do have a facility cost on the previous slide, which was about $4 million for installation.
Right.
But then these are yearly payments that are going to be made because of that facility.
Okay. That's all right.
So our profit margins, so we're going to be targeting our market, which is 20,000 patients that are diagnosed with GBM brain cancer. And so out of that market share, we're hoping to treat about a third of them, or 30%. So just based on our competitors.
Glioblastoma.
Yes.
Glioma.
Yes.
Okay, glioblastoma. Okay, that's an acronym.
Yes, so just based on our competitors, we're going to just price our drug roughly the same amount, which is $50,000 per treatment, which is going to bring our revenue, assuming we treat 30%, to $300 million per year. And then just minus our cost production, we get a profit margin, which is about $240 million. So.
Well, if you save the glioblastoma patients, you're a Nobel Prize quality. Although I'm hearing that it's it's producing miracles somewhat.
Yes. So
this immunotherapy.
Yeah. So this is going to be our net growth for our company outlook. So we're going to, as a pharmaceutical company developing a new treatment, we're going to spend quite a bit of time going through clinical trials and developing our drug. So this is just like the average time span, and these are just some estimates on the amount of capital we're going to have to spend to go through the FDA approval process. And these are the hits that we're going to take until we finish our phase 3 clinical trials, which could take up to 12 to 15 years. So it's going to take roughly $2 billion. So this is based on millions of dollars in United States.
AstraZeneca for one drug, $12 billion. So, and
Wow.
Amgen, $3 to $4 billion, and they were the most reasonable. So
Wow.
you're not far off.
Yeah. So just based on our revenues, and we're expecting to make roughly $200 million per year once we get this drug approved, and it'll take about 8 years actually to break even with our cost for approving the drug, and then after 12 years after the approval process, after 24 years, we're going to expect to make $1 billion.
I wouldn't call those trial costs capital expenses.
Yeah. Right. Yeah. This is a capital expenditure. We probably could have just found a better term for that.
Because capital usually has to do with fixed equipment.
Yeah. So we
But that's right.
we took in consideration the costs associated with the approval process, and also some of our variable costs as well, which isn't as significant as the approval process.
Can't do things perfectly.
Yeah, so for pilot plant safety analysis, it's pretty basic, so required lab and safety training, PPE required, gloves, goggles, lab coats. We'll be able to generate standard operating procedures, which would also include health and safety in them as well, and then emergency procedures, so just developing NFPA signs based on chemicals and just things like that. So we were able to create a hazard and operability study based on the various forms of equipment in the pilot plant. This is just a sample of a few of them. So two components are personnel and one piece of equipment. So for the personnel, it would just be plant contamination, which I characterize as low risk, just because as long as you have proper PPE and you're not wearing gloves into other areas, it's pretty easy to mitigate. And then equipment, one piece of equipment deviation could be dissolvent failure, risk being minimum. So raw material contamination into the feeder. If feeder requires extra energy input, electrical safety could be a concern. So possible consequences could be contamination of final products. And then, so a safeguard would just be control systems, which one which monitors electrical input into the dissolver, and then a second would be a way in which we continuously monitor like the material what's the word I'm looking for? Like the material composition, so that way we can continuously know whether there's been a deviation or something of that sort. And then recommendations would just be operator paying very careful attention to kind of mitigate contaminations, and also monitoring electrical power from through monitoring stations as well. So.
This is a sort of a risk analysis. There are other ways to do it, but
Yeah. This was just like it was like really long, so I just kind of
Okay.
So for regulations, we actually don't have to secure that FDA regulations for the nanoparticles that we're using because they're already FDA approved. So the FDA, I believe, has a rule in which that if the individual polymers you're using are already FDA approved, if you sort of put them together, then that product is also FDA approved.
Don't count on that.
Okay.
Because it's a new chemical. It could be viewed as a new chemical entity because of interactions of different components. So I would not count on that at all.
Okay, that's also a possibility. Yeah.
Especially if it's going to, part of that $200 million is getting what it takes to have FDA approval.
So for clinical trials, it would just be a 3-phase trial. So preclinical studies would be lab tests and manufacturing procedures, which is what we'd have in the pilot plant. And then small scale testing, so type of cancer effectiveness compared to previous cancer treatments. And the final scale of testing would be testing on actual people. So
how close are we now? Because we've
already go
to finishing.
Oh, yeah, I think it's just like the second to last. So
Okay. Because people are waiting and we had some trouble. But
yeah.
Okay. You have something?
Finish your slide.
Yeah. Next one. Yeah.
So in summary, we have proposed a technology for in situ gene editing of macrophages, which we believe could drastically decrease the costs of immunotherapies and bring it to a wider market eventually. We've also developed potential processes and economic analyses for production of the nanoparticles that we would use in this therapy. Some of our goals that we have achieved throughout this project are in researching potential targets for this gene editing therapy, developing a mechanism to recruit the immune response, as well as researching economic feasibility, potential processes we could use, and coming up with initial plans for a pilot plant.
Okay, so future development. For our company, we want to test the efficacy of nanoparticle delivery and gene editing in vivo. We want to redefine clinical indications and disease targets through our research and development. Further strategies for our plant deviation, continuous improvement on our facility, our processes, redefining our process approach, and also approaching venture capital firms to kind of seek additional funding for our process.
Thank you. Any questions?
Great. You have references. I was worried about that. Often references are on the slide, so people know where it comes from. But you absolutely have to have references. I was going to catch you on that, but I don't have to. Thank you so much.
Thank you.
Thank you.
Just me. Are there animal studies that I didn't notice that you're going to do?
That's
Animal studies Animal studies are usually, I believe,
in our preclinical
First phase.
trials.
Right.
Yeah. That's what this is referring to, the in vivo studies. Yeah, we didn't actually get around to doing any animal studies in the course of this project, but
you will have to do them.
Yes, absolutely.
If there's an animal model for a glioma, you will have to do the animal studies. Here's the one thing that people are waiting, but we did a study once, and it was on the uptake of hyaluronic acid into cells based on the CD44 receptor. And if the molecular weight of the HA was above, if the molecular weight was like 120,000, it was carried into the cell, into the nucleus, into the nucleoli. If it was 250K or larger, it attached to the CD44 but it didn't get carried into the cell. So if you're going to start a company, make sure you choose your HA of the right molecular weight. This is something that's not known that much, but it really is quite dependent on the molecular weight of the HA.
That's actually really good information. We've seen some research indicating that different weights of HA and chitosan as well, different polymer sizes, also can affect the composition of the nanoparticles. So we want to aim for nanoparticles of a certain size, and we'll have to tweak around the actual molecular compositions that we use in constructing those.
Remember that HA is water soluble, so it's going to have to act really quickly. If you cross-link it, then it could stop right at the cell wall, and so you have to have that HA, which is water soluble, do its job very quickly because it'll just wash right off. Make sure you keep that in mind. I have a feeling after working with Professor Webster that he's going to want to cross-link it, and then it's not going to get carried into the cell. So keep that in mind.
Yeah, so that's why in our process diagram, so I add the HA solution in the last.
You added the
HA solution in the last.
Well, it
to
has
make
to
sure,
also
yeah.
in the body. It has to not
Yep.
dissolve before it gets into the cell too.
So we have the chitosan.
You have
But the chitosan, the second
Supposed to
shell.
help with stability.
Yes.
The
the
chitosan.
chitosan forms a complex that will increase the stability, but it might not allow HA to go into the cell.
Okay.
We labeled it fluorescently in order to show that. Well, thank you all. I wish I had more time because it's an area that I'm interested in, but people are waiting. You did real well. You will get a good grade for this.
Thank you.
Thank you.
Thank you.